Morning, children. All are doing good, hope. And last session I completed till the blood pressure, and we discussed about types of circulation, heartbeat, cardiac cycle, blood pressure. All I completed in my last session. Today I am going to see some other few topics other than circulatory system but related with the circulatory system. Okay, what are the things they are? First one is instrumentation. What are the instrument? That is the instrument which is used to measure the circulation and working of heart and other health related issues. So those instruments are called stethoscope and sphygmomanometer. And next one is blood grouping. You know, children, we are all out having a same type of blood. Okay, so some are having A, some are having B blood group, some are having O blood group, some are having AB blood group. In that, some are having O positive, O negative, A positive, A negative, like that. So, and then this also we are going to see. And next one is blood transfusion. So what is a blood transfusion? In under this topic we are going to see how to transfuse the blood. If is in can anybody can transfer the blood to any person or not, or what is the procedure for that? And then next last topic will be the lymphatic system. Next is a new topic for you children, a new system also for you. So in our lower classes you have studied about a digestive system, circulatory system, reproductive system, excretory system, all. But here this lymphatic system is entirely new for you, right? So okay, it is nothing but what is lymphatic system? It is nothing but the lymphatic system. It is like a circulatory system, another one circulatory system or transport medium in our body. Okay, right? And like blood, it also has plasma. You know, I said blood consists of liquid part and solid part. The liquid part is plasma, the solid part is what? Blood cells. Same way, in lymph also it consists of plasma like blood. The composition of plasma is same as like blood. But the regarding blood cell, if we take, it has only one type of blood cells, that is lymphocytes alone. No other blood cells are present, right? So then what is its main function? It is nothing but, see, it has a main function. Since lymphocytes are there here, it is involved in the defense mechanism of our body. You know, children, lymphocyte is a type of white blood cells. This lymphocytes involved in the antibody production and destroying the infected cells. Since it produce antibody and neutralize the activity of pathogen and destroy the infected cells, it involved in the defense mechanism of our body. So this is the functions of what? Lymph. We can talk generally. So in overall view, if we say lymphatic system involved in the defense mechanism, that is its main function, and apart from that it do some other function also, that all we are going to discuss now. Okay, so let's enter into the topic, children. Stethoscope. A stethoscope is used to detect the sound produced by the internal organs of human body, right? Stethoscope is not only used to find the heartbeat, it also used to find out the functions of internal or sounds produced by the internal organs like stomach, liver, kidneys, so like that. So these are the internal organs. These organs also produce some vibrations in it. Okay, so to find out the internal organ, the heart sound is heard by placing the stethoscope on the chest. Okay, when we are not feeling well, we are going to doctor for treatment, isn't it, children? What the doctor immediately do? He keep a stethoscope on the chest and observe the heartbeat sound as well as internal. If the internal organ is not functioning, that also we can find. Okay, then it is useful diagnostic tool to identify and localize health problem and diagnose diseases. So this stethoscope used to find out the diagnosis of diseases and any local health problem also we can find out. The modern electronic stethoscope are high precision instrument. Okay, that's all. The diagram is given, please note it. And this next sphygmomanometer You know children, sphygmomanometer, this is also very familiar for you. Whenever you are going to any hospital, then with your parents or any elders, or occasionally you can watch the measuring of blood pressure, isn't it? So the instrument which is used to measure the blood pressure is called a sphygmomanometer, right? Sphygmomanometer also a clinical instrument. Like that, many clinical instrument nowadays used in the hospital. Okay, so sphygmomanometer is a clinical instrument used to measure blood pressure when a person is in a relaxed and resting condition. So please note down the point children. When the blood pressure should be measured in a relaxed and resting condition of a person. If we want to get a blood pressure, we should be in resting and relaxed state, then only we can take a blood pressure. Suppose if you are if you went to play, or if you went for exercise, or soon after the exercise, soon after the play, if you take the blood pressure, it shows abnormal measurement. So okay, so we cannot consider that. Same way we cannot take blood pressure during the sleeping time also. So when you are relaxed and resting condition, the blood pressure should be taken. The pressure of the brachial artery is measured. It helps to estimate the state of blood circulation and working of the heart. Okay, what is the by measuring the blood pressure, what are the things we could know? That is, we could know the circulation of blood, how the circulation is going on inside of our body, whether it is going on properly or any disturbance in that also we can find. Okay, and also the working condition of the heart. So we can find that the heart is working properly or not. Okay, so this is the functions of sphygmomanometer. By using sphygmomanometer, we can find out the blood circulation and also working of heart. It helps to diagnose continuous conditions such as increased or decreased blood pressure. And what are the instrument, what are the type of sphygmomanometer? One is monometric, another one is modern digital type. Usually in the hospitals also mostly they are using monometric only. Nowadays some patients in a family, one or two two persons, if have high blood pressure or low blood pressure, they are getting modern digital sphygmomanometer by their own. They themselves measure the blood pressure. Okay, so these are the two types of sphygmomanometer. One is monometric, another one is modern digital type. Next one is next topic what we are going to see is what blood grouping. So you know children, already I said there are different types of blood groups we can see in humans. Okay, there are A, B, AB, O blood group. These are the four types of blood group, major type. Apart from this A1 positive, A2 positive, that all are what subdivisions of them. Okay, so the main four types A, B, AB, O. Okay, who first discovered these blood groups? That is another question. Okay, see, Carl Landsteiner developed the blood grouping concept. Okay, he identified the blood groups of A, B and O. Then AB blood group was recognized by Degastelio and Steiny in 1902. So Carl Steiner first observed only three types of blood group, that is A, B and O only. So later on in 1902, Degastelio and Steiny discovered or recognized another one blood group, that is AB. Okay, next one, how to identify this blood group. Okay, human blood contains certain specific substance called agglutinogens and or antigens, and agglutinins or antibodies. Okay, so if we take a blood of any human, we can find two things in the blood. One is in the plasma, another one is in the surface of antibodies. One is in the plasma, another one is in the surface of the antibodies. What is present in the plasma? That is antigen, that is agglutinogens or antigens present in the plasma. Then what is present on the surface of RBCs? Antibodies or agglutinins are present on the surface of the antibodies. Based on this antibodies and antigens, we can classify the blood group. See children, if the blood group, if a person having a blood group A, their antigen also A, but the antibody is ulta, that is B antibody will be there. Okay, same way if a person have B blood group, in its plasma they have what? Antigen B, and the antibody on the surface of RBC is what? Yes, right, A. Like that, based on this agglutinogens and agglutinins, we can classify the blood group. Okay, antigens are found, see, come for the text children, antigens are found on the membrane surface of the RBCs. Antibodies are, ulta I said, antigen present on the surface of the RBC, antibodies are present in the blood plasma. Based on the presence or absence of antigen and antibodies, human blood group is classified into 4 groups, that is A, B, AB and O. An individual has one of the 4 blood groups. Okay, so if a person has A blood group, A group individuals have antigen A is present on the surface of RBCs, antibody B is present in the plasma. Then if a person have B blood group, he has antigen B is present on the surface of the RBCs and antibody A is present in the plasma. Then okay, what about AB blood group persons? So they have both antigen A and B on the surface of the RBC, but there won't be any antibodies found in their plasma. Okay, excuse me children, sorry. Next, O blood groups, it is also ulta to the AB blood group. They don't have any antigens on their RBCs, but they have both antibodies in their in their plasma. Okay, so that's what they are trying to see. See, once again we recall this children. A group individuals having antigen A on the surface of antibodies, antibody B in the plasma, and B blood group individual has B antigen on the surface of antibodies and A antibodies in the plasma, and AB blood group individuals having both the antigen A and B on the surface of antibodies, and no antibodies are present in the plasma, right? And then next, O blood group, there is antigen A or B are absent on the surface of RBC. However, the plasma contains both the antibodies A and B. Okay, so that's it about identification. Based on this, we can identify the blood grouping. Okay, next, blood donation. So this regarding this ABO blood group, you are having a chart in the next page, we will see that also. So come for a blood donation. In blood transfusion, one must consider the antigen and antibody compatibility between the donor and the person receiving blood. Okay, that is recipient. When an individual receives a mismatched blood group from the donor, agglutination of blood occurs in the body, which leads to the death. Person with AB blood group are called a universal recipient, and they can receive blood from person with any blood group. Same way, person with O blood group are called a universal donor, as they can donate blood to a person with any blood group. Okay, right? So children, regarding blood donation, before blood transfusion, we must consider the antigen and antibody compatibility. Antigen and antibody compatibility of both donor and recipient. Both donor and recipient. If the blood group will be differ, if the same blood group, no problem. So donor also A blood group, recipient also A blood group, no problem. There won't be any problem, the recipient body, except that blood group. But if a donor is A and the recipient is B, what will be happen? There will be a agglutination. There will be a agglutination or coagulation, blood getting clumped, and it leads to what? Death. Okay, so that's what blood donation. During blood transfusion, we should consider this screening of blood. And then person with AB blood group. Next come, who all given blood to whom? So AB blood group, as commonly said as what? Universal recipient. Why they are saying as universal recipient? They can receive blood from any of the person blood group. Okay, AB blood group can receive blood from A blood group, O B blood group, O blood group, AB blood group also. But he can give only to AB blood group only. He can give blood donation to only AB person. He cannot give to all those person. So that's what he received blood from all the type of blood grouping person. So they are called what? Universal recipient. But he can donate blood only to the particular AB blood grouping only. Then next one is universal donor. Who is universal donor? O blood group is called universal donor. Why they are called so? Because the O blood group can give blood to all the type of blood group persons. Okay, he can give to O, he can give to A, B, AB, all. So since he can donate blood to all blood group persons, it is called as O blood group is called as what? Universal donor. But he can receive blood only from O blood group person only. He cannot receive from any other persons, right? That you should keep in your mind. And then next, RH positive. What is RH positive? Another one question. Okay. So when you are taking blood grouping test, they will say O positive, A negative, AB positive, like that they will say positive, negative. Okay, this is called what? RH factor. This is called what? RH factor. So see, RH factor first discovered by Landsteiner and Weiner only. From which animal they have first discovered? From a rhesus monkey they have discovered first. That's what the factor is called what? RH. RH is a first two letters of rhesus monkey. Okay, so then the surface RBC contain antigen for RH factors, that is RH positive. Person have RH antigen on the surface of RBC while RH negative. So what they are, the surface of RBC contains the antigen for RH factor. So they are called what? RH positive persons. Person have RH antigen on the surface of RBC while RH negative persons do not have RH antigen on the surface of RBCs. So antibodies developed against this RH antigens is called what? RH antibodies, right? So if a person have RH antigen on the surface of the RBC, they are called what? Yes, right? RH positive. If they don't have antigen on the surface of RBCs, RH antigen on the surface of the RBC, then they are said to be what? RH negative. So the antibodies developed against this antibodies is called what? RH antibodies, right? Okay, children. You can see the table. See blood group A, antigen A. See the table. A blood group, the antigens on the RBC is antigen A, antibodies in plasma is what? B. Can donate to A blood group person can donate blood to A person and AB person and can receive from A and O. Okay, and then B blood group person, they have antigen B on the surface of RBC and anti-A in the plasma, and they can donate blood to blood group B and AB and can receive from B and O. And next one is AB blood group. Antigen present on the surface of RBC is antigen A and B. They don't have any plasma in their antibodies. They can they can donate blood to only AB blood group and they can receive blood from AB and O. So hence it is called universal recipient. And next one is blood group O. They have no antigen on the surface of RBCs. Then both the anti-A and B are present in the plasma. Can donate to A, B, AB and O. So it is called as what? Universal donor, and can receive blood from only O, right? Okay. Next, lymphatic system. The last topic of this lesson. Lymphatic system. The lymphatic system comprises of lymphatic capillaries, lymphatic vessels, lymph nodes and lymphatic ducts. Okay, so what are the organs involved in the lymphatic system are lymphatic capillaries, lymphatic vessels, lymph nodes and lymphatic duct. Lymph is the fluid that flows through the lymphatic system. Lymph is the fluid which flows through the lymphatic system. Like in blood vessels, blood is flows through, no, the same way in the lymphatic vessels, lymph flows through. And then lymph. What is lymph? A lymph from the intercellular space drains into a lymphatic capillaries. Lymph is a colorless fluid formed when plasma, proteins and blood cell escape into the intercellular spaces in the tissues through the pores present in the walls of the capillaries. Okay, when the limbs are formed. See, you know very well children, in the blood capillaries, blood is circulated, heart pump blood to the aorta, aorta to arteries, arteries to arterioles, arterioles to meta-arterioles, meta-arterioles to capillaries. See children, capillaries are very thin and transparent, single layered wall. So through this capillaries, plasmas, proteins, some plasma, some protein and some blood cells are escaped. Okay, so then it is enter into the intercellular spaces. These plasma, proteins and blood cells are again enter into the lymphatic capillaries, and from the lymphatic capillaries, these together, the plasma, protein and blood cells together form a fluid called lymph. The lymph enter into the lymphatic capillaries from intercellular spaces. From the lymphatic capillaries, it is enter into the lymphatic vessels. Okay, right? So now you clear how the lymph are formed. Okay, present in the walls of the, it is similar to blood plasma but is colorless and contain less protein. When compared to blood plasma, the protein content is less in lymph. The lymph contains very small amount of nutrients, oxygen, carbon dioxide, water and WBC alone. No RBC, platelets are described here. Okay, next we are going to see about functions of lymph. What is the functions of lymph? See, it is a 4-mark question children. Function: supply nutrient and oxygen to those parts where blood cannot reach. Okay, you see children, blood cannot reach all the regions of our body. So what happen in those regions? The lymph take over the functions of blood. There the lymph supply the nutrients and oxygen those areas. So then it drains away excess tissue fluid and metabolites and returns protein to the blood from the tissue space. As I said, from the capillaries, the plasma, some blood cells, some proteins are oozes out. These substances are collected by the lymph and again returned to the blood from the tissue spaces. Okay, and then the lymph also carries absorbed fats from a small intestine of the to the blood. Okay, so after digestion, the food are converted into their respective simple substances like carbohydrate into glucose, protein into amino acid, fat into fatty acids and glycerol. So in the intestine, small intestine, the food digestion gets completed and the end product of glucose, amino acid and fatty acid glycerols are formed. The glucose and amino acid absorbed by the blood capillaries of the villi. And about what fatty acid and glycerol, it is absorbed by the lymph gland, absorbed by the lymph in the villi of the small intestine. So that's what they are saying. The small lymph also carries absorbed fat from the small intestine to the blood. The lymphatic capillaries of intestinal villi absorb digested fat, and then lymphocyte in the lymph defend the body from infection. So these are the 4 functions. That is, it supply the blood where blood can it supply nutrient, oxygen where blood cannot reach. Next one is it drains away the excess fluid and metabolites and return protein to the blood from the tissue space. Next one is it carries lymph carries absorbed fat from the small intestine, and since the lymph consists of lymphocyte, it involved in the defense mechanism of the body. These are the 4 functions of lymphatic system. Okay children, the lesson is over with this topic. So okay, what we have discussed in this lesson are transportation in plant, circulation in animal. In transportation in plants, we are seeing the different types of transportation like diffusion, active transport, osmosis, imbibition, all we discussed. Okay, and pathway of absorption of water, all we discussed. And then regarding blood circulation, we have discussed many things. Okay, composition of blood, then structure and working of heart, and types of circulation, types of blood vessels, heartbeat, cardiac cycle, blood pressure, and the instrument to measure working of heart and blood pressure like a stethoscope and blood, the sphygmomanometer, and blood donation, how to transfuse the blood to a recipient, and our H factors of the blood, then last lymphatic system. These are the topics we were discussed in our last classes. Okay, so children, hope you understood about this lesson, and these are the 3 lessons. What are the lessons I have taken for you? 3 lessons I have completed to you. Okay, first 3 lessons. So plant physiology and organization of organism, and this one, so the transportation in plants and circulation in animals. These are the 3 lessons which are the portion for the quarterly examination. Children, already I sent a PDF for the book back of this lesson, right children? So if you won't get any PDF to the school, and they have taken xerox and given to you also, I think so. You might be collected from the school. So anyhow, I sent the 3 lessons PDF along with 1 mark in the group. You please, any clarification you can clarify with that PDF children. So start prepare for a quarterly examination. Do your quarterly examination well. Okay children. Okay, we will meet in our next session. Bye. Thank you.